bunch of people online as well. So thanks very much for coming out. It was great attendance. I think it's a success of a joint CS QME initiative here. Thanks very much for coming out. So it's my great pleasure to introduce Uthsav Chitra. He's currently a postdoc at the Broad in the Eric and Wendy Schmidt Center up there. Prior to joining the Broad, he recently received his PhD in computer science at Penn University under the mentorship of Ben Raphael. His PhD thesis was algorithms for understanding the spatial network organization of biological systems. Prior to his PhD, also received an SCB in mathematics, an AB in computer science, and an AB in applied mathematics from Brown. In fact, I just learned that's where he first met Ben Raphael back at Brown. He also spent a year at industry at Facebook working on some of the ad systems. His research broadly develops rigorous statistical and machine learning methods to address fundamental questions in biology with a focus on problems involving spatial structures or graphs. His research has appeared in several top computer science conferences, including one as of 2 minutes ago that we can go here a little bit about, as well as a number of top computational biology journals. His work has been recognized quite broadly with a number of awards, including the Rising Stars in Data Science Award, a Best Paper Award at Recom, a single scholarship, and an NSF graduate research fellowship. I'm very excited for his talk today on machine learning for spatial and network biology.
Yeah, thank you very much, Mike, for the introduction. Really excited to be here. So, as like I mentioned, I'm Uthsav Chitra. I'm at the Broad. I recently was at Princeton, and today I want to share with you some of my research on. My research on machine learning for spatial and network biology. So to start off with some background. So to start off with some biological background. Sorry. You must just have to stand up this flat. Okay. Okay. So biology is characterized by interactions. Biology is characterized by interactions. At the small scale, we have genes, which encode proteins. These genes and proteins interact with one another, forming these large and interconnected networks of these gene and protein interactions. At the same time, many of these gene and protein interactions often occur within individual cells. And we have many different types of cells in our body that interact with one another, forming these cellular interactions. Moreover, these cells interact within the different tissues and organs in our body. And so we get these spatial interactions between different cells, which can lead to different organizations of tissues. Understanding these interactions is really important for understanding human health and disease. So for example, in cancer, oftentimes mutations will alter a small subnetwork of these gene and protein interactions. And so, for example, there's many gene or protein subnetworks that are altered by cancer and recur across many different patients. For example, I'm showing one example here. At the same time, in cancer, these alterations also affect the interactions between cells. And this can disrupt these spatial interactions and lead to different organizations of tissues. For example, you can see the normal cells in cancer, they kind of like, you have this uncontrolled growth. And so in my research, I very broadly developed computational and machine learning methodologies for understanding these biological interactions, both at the network level, where you have interactions between genes and proteins, and at the spatial level, a bit hard to see, but where you have like interactions between cells. And moreover, I also study how disease can alter both these network and spatial interactions. There are many algorithmic challenges in doing this work. Many of our biological measurements tend to be very noisy or limited. For example, our knowledge of interactions between genes and proteins, the interaction networks are often incomplete or just unknown. And the way that we can measure cellular interactions, we often get very sparse data. So it can be very hard to even determine which cells are interacting with each other. And moreover, I've shown you these very nice pictures, but in real data, we often have very large and high-dimensional data sets. So for example, networks of genes and proteins have tens of thousands of genes, of nodes with hundreds of thousands to millions of edges. And inside tissues, you can have thousands to even millions of individual cells. And so because of these limitations, oftentimes existing computational frameworks don't map on very neatly to biological problems. And so you need new computational methods for biology. And so in my research very broadly, I developed these different computational methodologies for biological problems. This includes methodologies based on statistical inference for studying networks or geometric methods for looking at spatial interactions. And in my work, I developed both mathematical theory and practical algorithms when appropriate. And I've used these algorithms to understand important biology. For example, the gene and cellular interactions that underlie diseases like cancer. And this often involves collaborating with experimentalists, for example, neuroscientists or chemists, and by doing large-scale analyses, for example, looking at data from large cohorts of patients with cancer. And so today I wanted to talk, and I guess these images are not super bright, so hopefully you can see them. But I wanted to talk about two lines of work that I've been very excited by past several years. One is on modeling spatial variation of gene expression, and the other is on identifying these gene subnetworks that are altered by disease. And I'll very briefly mention at the end some of our work on inferring these gene interactions. And so I'm going to start by talking about our methods for looking at spatial biology, spatial variation and gene expression. This is joint work with a number of very helpful co-authors, including my co-first author, Uthsav Chitra, and my PhD advisor, Ben Raffert. So the way that we study spatial biology is through a technology called spatial transcriptomics, which at a very broad level, you can imagine you take a thin slice of a tissue, for example, a brain or a liver, and at different spots in this tissue, hopefully it's not too hard to see, but at these different spots, you can measure both their spatial coordinates, the x, y, and you can measure something called gene expression, this very high dimensional vector. And so you measure thousands of genes, up to 20,000 genes, and even thousands to millions of these spots. And so what you get in terms of data is you get this matrix of spots by gene, as well as these 2D spatial coordinates. And since this is at least partially a computer science department, I want to kind of contrast this type of data with data that we might often see in machine learning or, say, computer vision. And so one of the kind of interesting computational aspects is that you get both high dimensional data and you have spatial data. So for example, if you're working in machine learning or high dimensional data analysis, typically you don't also have this spatial information. Whereas in computer vision, you might have, in terms of pixels, you can get kind of like 2D spatial coordinates, but typically your features are low dimensional. You'll measure like RGB. And so here, there's very interesting computational challenges figuring out how to model both this high dimensional structure of these different genes and the spatial structure. And so what are some things that you can do when you have this kind of spatial high dimensional data? One thing you could do is a very simple task of clustering. You can imagine you take these different spots and you try to find spots that are close by in space and have similar gene expression vectors. For example, in the brain, these different clusters might correspond to these layers that you can see are stacked on top of each other. Or in a tumor, these clusters are like, this is the tumor, this is like surrounding normal cells, the lymphoid, and like all these other interesting cell types. Another thing you can do is you can look at individual genes. And so this is kind of like a column of this matrix. And if you look at this column and you visualize it in space, you can try to find these like spatial patterns of these gene expression values. For example, this gene, it kind of slowly increases and decreases in space. There's this gradient of gene expression. And another thing that you can ask is if you can also try to understand how cells interact with one another. And typically you do that by looking at pairs of genes that are known to mediate these cellular interactions. For example, these ligands are released from one cell and they get received by another cell. And by looking for correlations between columns, you can start to understand how cells interact with one another. Unfortunately, what makes all of these tasks really hard is that the data in general is really sparse. And so this matrix that I've shown you of spots by genes tends to have 70% or more zeros. And so in practice, what this means is that most genes kind of look like this picture on the right, where it's a bit hard to see, but at these different spots, most of them are zero. There's a few ones and twos, but in general, the dynamic range of your measurements is very low. And so today I want to talk about how you can overcome this sparsity by modeling the spatial structure of your data. And in particular, I'll talk about how you can model spatial structure in order to understand tissue organization, these different clusters, and to find these spatial patterns of gene expression, like these gradients. And at the end, I'll talk a bit briefly about our ongoing work on modeling interactions between cells. And so how can you leverage spatial information to overcome this sparsity? So one of the standard approaches that's often used in the area, most methods rely on this kind of locality assumption. The idea that if you have a location in space, it's like nearby spots should also be kind of similar, and you can aggregate measurements along this like local neighborhood to help account for sparsity. So for example, methods that kind of implicitly use this local assumption include things like graph neural networks or hidden Markov random fields or Gaussian processes. But in many tissues, you actually have a global structure. So for example, this cortex that I'd shown before, you can kind of see that there's like this layered 1D structure. You have these layers that are like stacked on top of each other. And in our body, we have many layered tissues. For example, the eye has layers, skin has like 3 layers, your bone has like kind of concentric layers. But these local methods often like can't describe this global geometry. So for example, here's one output of one of these kind of local clustering methods, and you see it's not totally capturing this like 1D structure. And so a couple of years ago, I was kind of interested in looking at this spatial data. And when I saw this like 1D cortex, we were thinking that we couldn't find any models for describing this global 1D geometry. And so we had to come up with a new model. And so being kind of mathematically minded, it's kind of easy to start with the simplest case. So if we're interested in layered tissues, what is the simplest type of layered tissue? This is like not showing very well. But you can kind of see, I guess, that in the simplest case, you can imagine your layers are the like horizontal strips. They're all parallel to one another, and they're axis aligned. Here's that. And so in the simplest type of tissue, if you really believe this layered geometry, then you should believe that your measurements only depend on this x-axis and that they're constant along the y-axis. And this is because all of these points on this y-axis are equidistant from these layer boundaries. And so this sounds like a simple assumption that gene expression only depends on this x-coordinate, but the reason it's really nice is that even in 2D, when you might have very sparse gene expression measurements, by kind of aggregating gene expression along these y-axis points, all these points with the same y-axis, you can find these very dense patterns in 1D. And so for example, for this gene, this is a real gene, you can see there's like jumps at the different boundaries of these layers. And there's also these kind of smooth gradients in between each layer. And so one very simple model that you can use to describe this kind of data is with a piecewise linear model, where you have these pieces that describe these different layers, and then these gradients describe like smooth variation within a layer. Of course, you could also use more complex models, but just to kind of start simple, a piecewise linear model seems like a fairly simple way to describe this type. And so this is like the simplest, oh, sorry.
Okay. I have a question about the sparsity of the data. In that plot or in that figure there, is each point a cell or is each point a gene like sample?
Yeah, this is a good question. Not too clear. So each point here is a cell, and I'm coloring it by it's like shaded or it's like less shaded if that cell is expressing a specific gene. I forget which gene it is.
Okay,
like
so the sparsity in the data is from sampling only few cells, or like I guess.
It's more that every cell has like, if you sample a bunch of these cells, here it's spots, it's actually a combination of cells. But for most genes, it's 0 inside that cell, just because you don't measure, you don't capture, like your sequencing is not really high, so you're not capturing a lot of it. Yeah. That's like the main driver, or one of the drivers of the sparsity. Yeah.
So the organization will mostly depend on in which plane are you getting the cut of the tissue, right? And,
Yeah.
So this expression kind of mapping will be there for all particular, like all of the genes in your codex of the genes that you're looking for, like the expression of that particular gene you're looking for.
I understand that you're asking if you can measure all.
So on the y-axis, when you mention the expression, do you mean to say how much of that particular gene is being expressed in that location?
Yeah, so this is like per gene. And you can imagine you can have each a plot for like different genes. But this x coordinate, this like kind of layered from the spatial organization of the tissue. And this is again like kind of the simplest example. So I'll talk a bit more about how you can like kind of learn this geometry from more complicated tissues. Where yeah, you're right. Depending on the cut of the tissue, it might not have this like very simple structure. Yeah, so this is kind of like the simplest case. And maybe a next natural question is like, what about for more complicated cases? So for example, for many layered tissues, you can imagine these layers kind of curve in space, they have arbitrary shapes. And so ideally what you'd like is you'd like a way to transform each of these kind of curved layers into the simple case from before, these axis aligned layers. And you want to find a transformation that's like somehow smooth. It's not like very obvious. And also preserves, okay, this is really hard to see, but if you like squint, you can kind of see these grid lines that like, you want to preserve these grid lines, which tell you this like layer depth axis. Maybe it's a bit easier to see on that one. But yeah. And so it turns out actually, there's a very nice way to do this using complex analysis, using something called a conformal mapping, which is a map basically between two regions of the complex plane, which you can just think of as two regions of space that is both smooth and it preserves angles between any two curves. And so in this case, it preserves these 90-degree angles between these grid lines. This might all sound very abstract, but if you want to think about this concretely, you can also compute these maps by solving something called the heat equation, where you place heat 0 on one side and heat 1 on the other side. And then these lines of equal heat, which are called isotherms, give you this layer depth axis. And so putting these two ideas together, you can pose a very general problem formulation for finding this layered structure, where the idea is that, sorry, two questions.
When you transform the data from like this nonlinear to this like linear form, are there any issues with like artificially sparse or dense areas? Because it looks like, you know, some areas that might correspond to like a more curved region are now straight, and some that are maybe a less curved region are close. Like if something's like this and you make it straight, it's going to be more sparse. That makes sense.
I kind of, yeah, in general, like, I guess you can't find a way to like totally, like if you want to interpolate between two curves in some sense, like there's always going to be some regions that are like, and the curves are not like equally spaced apart. And there's always going to be some like difficulties in doing that kind of way.
Do they balance out, or do you just kind of have to acknowledge that going forward with your analysis?
Yeah, they might not necessarily. I guess, so in some sense, it sounds like you're asking these lines of equal heat, they might not all contain the same number of like points inside each line. And yeah, depending on the geometry, that might be the case. I'll show some pictures on like real data and also describe how we actually do this. And then maybe if you still have a question, circle back. So the general problem that we'd like to solve is that given this kind of data, this spatial data where you have these plots and gains and you have these 2D spatial coordinates, I would like to find these conformal maps, these like layer specific maps, and these piecewise linear functions that best fit my data. And there's kind of natural ways of how to define best fit, but that's not too important, so I won't get into the details here. It turns out though that this problem formulation, although we could form it, is actually a very hard problem to solve. Because in particular, optimizing a function over a bunch of different formal maps in like arbitrary areas is, or at least for us, turned out to be too hard, at least at this moment. And so we were able to solve two special cases that were still pretty useful for real data. And so one of the special cases is if you have some measure of approximate layer boundaries, then what you can do is for every pair of these approximate boundaries, you can construct this conformal map by solving the heat equation. And this gives you this one coordinate. And then to find these piecewise linear functions, once you have this 1D coordinate, there's a dynamic programming algorithm for finding piecewise linear functions that best fit your data, which is something called segmented regression. The other case that we're able to solve is if you know your layer boundaries are lines, even if you don't know them, you can actually solve it using another dynamic programming algorithm. And this algorithm, at a high level, I'll say it's very similar to kind of the classical Newsnop's algorithm for RNA secondary structure folding, which you might have seen in bioinformatics. And so putting this together, we put this in an algorithm that we called the layer, where you simultaneously find these conformal maps and these piecewise linear functions that tell you both the layers of your tissue and these piecewise linear patterns of spatial variation in your gene expression measurements. You might be wondering where this name came from. Part of it's supposed to be a play on words because we're looking at these layered tissues. And then I also like to go rock climbing. So we'll see in this and a few other things I try to insert references to rock. But just to show you what this looks like on some real data, here is this region of the brain that I'd shown before. It's called the human dorsolateral prefrontal cortex. And we show that through this kind of global 1D model, we can identify these layers of the tissue more accurately than existing approaches. And also kind of just as interestingly, like once you have these layers, the kind of cool thing that you can do is you can actually look at gradients of expression using this 1D layer depth coordinate. And so, for example, these gradients might tell you, oh, this gene's kind of increasing or this gene's decreasing in this layer. And it turns out to be a good signal for what are called marker genes, which are just genes that are kind of important for biological function. And it can reveal both known marker genes and potentially new markers. And importantly, I want to stress that these kind of patterns of gene expression, these gradients, are often not identified by these local methods that don't take into account this global 1D geometry. And so to summarize, this is our algorithm per layer. We published it a couple of years ago at RECOM and here at Cell Systems. And using this geometric model of these conformal maps and by solving this heat equation, we could identify this layered structure and these gradients for tissues like in these different regions of the brain. And so this is really cool as we got to use a lot of kind of math that I hadn't really touched since undergrad. But one of the major limitations is that this approach requires you to have some prior knowledge of your tissue. So for example, you have to know some of these manually annotated boundaries, or you have to know that your tissue, these boundaries are like given by lines. But oftentimes in biology, we want to not just like, we also want to discover new biology. You might have tissues where you have very complex or unknown boundaries that you can't model with this kind of supervised approach. So for example, here, this is a region of the brain called the cerebellum. It also has these layers, but these layers are kind of discontiguous. The yellows don't connect. They kind of bend really wildly. And this kind of tissue is one that we couldn't study with our previous approach. And so as I was thinking about how to solve this more general problem, I noticed, so this was around 2022, 2023, everyone in my department was using deep learning for everything. So we had like ChatGPT and DALL-E, like all these things were really blowing up. And so I thought, I have this layered tissue problem. I showed you this problem from before. Maybe I can use deep learning to solve at least my problem. It seems simpler than, I don't know, loading up ChatGPT. And so it turns out actually that you can use a deep learning approach and it can learn this 1D structure, not just for these kind of tissues where we know something about them, but also these more complex tissues where it has these kind of curved and discontiguous layers. And so I want to describe this approach briefly, but I first actually have to take a step back because in order to describe what this deep learning approach is doing, it actually requires us to think a bit more high level about what it means to model spatial variation in high dimensional data. And so, okay, sorry, I'm going a bit back to math, but the natural way to model spatial variation is through the language of multivariable calculus. So in general, if you have a function in space, so f of xy, in our case, we have a function f of xy, where at every point, you have this high dimensional gene expression vector. It has these component functions f1 to fg, where g is your number of genes. And if you allow for discontinuities at different boundaries, that's not too important. But in general, if you have a function in space, the way that you model spatial variation is there's something called a spatial gradient, which is just the gradient of each of these individual functions for each individual gene. And so you might recall from multivariable calculus, the gradient of a function tells you the direction of maximum increase of that gene. And so, for example, it might be a bit hard to see these, but for this gene in blue, for example, the gradient here points in this direction, telling you that in this direction of the tissue, your gene is increasing the most. And for this other gene, this gradient in green, this gradient points in a different direction, telling you this gene 2 is increasing in a different direction. And then gene 3, it might actually be increasing in like the opposite direction. It could point in this way. In general, these gradients form a vector field across your entire tissue. So if you'd like to model spatial variation, your goal should be to learn these kind of gradient fields. Unfortunately, as I discussed before, this data is often very sparse, and so it can be hard to learn these gradients from your data without placing some assumptions on them. And what we found actually is that under a few simple assumptions, you can actually learn these spatial gradients. And so the assumptions, so there's two assumptions that we have. One is that these spatial gradients all point in the same direction. There's a single vector field, this bold-faced V, where the gradients for each gene are all pointing in that kind of plane in space. And notice that these gradients might have different magnitudes. For example, gene Schmidt, you can kind of see that gene 1, which is blue, is smaller than the gradient for gene 2, which is green, and gene 3 points in the opposite direction. But there's a single shared vector field, bold-faced V, that's like shared across all of these genes. And so that's the first assumption, that there's a shared vector field. And then the second assumption is that this vector field V is something in physics called a conservative vector field, meaning it doesn't curl around in space. And so this is a kind of intuitively a way to generalize this layered assumption, because in a layered tissue, your layer depth axis also shouldn't really be curling back like into itself. And so it turns out that this assumption that this vector field is conservative is equivalent to this vector field being the gradient of something called a scalar function, just a single function that at every point gives you a number. And this function in physics is sometimes called a potential function. So you have things like gravitational potentials or electric potentials. And so the cool thing, though, is that under these two assumptions, you can actually prove that your gene expression will have this 1D structure, that we assumed in our previous method of the layer, which is, and another way to state that is that your functions is f sub g for each gene, even though it's a function of two coordinates, x and y, your xy coordinates, really under these assumptions, this function is always a function of a single spatial coordinate, this d of xy, and then there's some gene-specific function. And so this generalizes this model from Belayer, where we model this d function, this using conformal maps, the heat equations. And so heat is one example of a potential. You can kind of see it with the colors, it's like white and dark red. And in heat, these lines of equal heat, these are called isotherms. These are these lines of equal thermal energy, I guess. And then for our coordinate, kind of motivated by that, we have these lines of equal depth. And so we call these coordinates an isodepth. And so we have this analogy also that this isodepth coordinate gives you a quote unquote topographic map of the tissue. If you squint again, you can kind of see, for example, many of us use topographic maps when we're hiking. These are maps that give you the elevation at every point in space. And then you have these like contour regions that surround different mountains or valleys. And similarly, we have this coordinate of a tissue where you can tell these arrows, these gradients tell you the direction of maximum change. Similar to if you're hiking, you can find out what are the steepest and flattest directions. And these contours of equal isodepth, the isodepth contours give you these like landmarks in space, these like domains or cell types or whatever you want to call them, how the tissue is organized. And so with this general model, you can pose a kind of problem of learning this isodepth coordinate from data. Again, you have your spatial data, this like gene by spot matrix and these 2D spatial coordinates. And what you would like to learn is you'd like to learn this isodepth function, which you have to assume is continuous and differentiable because you're taking the gradient, and these piecewise linear functions, again, that best fit your data. And what you can note is that this problem formulation, which we call the gene expression topography problem, is a relaxation of our previous problem, this problem for layered tissue, where we relax the assumption that instead of having these conformal maps, you actually just relax it with and you instead want to find a continuous function. And so one of the nice things about having relaxations of problems is they suggest better approaches for solving a problem. And so here we have a problem where you want to find two functions that maximize a certain likelihood, this like functional thing. And in general, if you want to find functions, you can approximate functions with neural networks. The neural networks are known to be like very good approximators for functions. And if you kind of work through the math, I won't go through kind of the details here. It turns out if you assume that these isodepth and these like piecewise functions are neural networks, this is actually equivalent to training a very specific kind of neural network. So this is a neural network where the input are your spatial coordinates x and y, and your output is your gene expression. But this neural network had one hidden layer in the middle that has a single node. And so this single node is what we call our isodepth function. And you can sort of think of this like an autoencoder in some way, where you start out with some input, here it's 2D, and we're trying to compress it down to 1D. So you at least have this dimensionality reduction from 2D spatial coordinates to 1D. And this neural network has to learn a 1D coordinate that best fits your data. And this is something we didn't know until like after we kind of published this, or at least submitted this paper, is that this kind of model where your inputs are spatial coordinates and your outputs are measurements actually has a name in the machine learning literature. It's called a neural field model. So you might have heard of models like NERF, which kind of use a model also where inputs are spatial and outputs are kind of observed measurements. And so just to kind of put this all together, we put this in an algorithm that we call Gaston, which is a backronym for gradient analysis of spatial transcriptomics, blah, 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 something that spells Gaston. And I came up with that backronym though, that the exact like words aren't that important, but this was supposed to also be a rock climbing reference. Gaston is a type of move when you go rock climbing. However, when I tell people about this name, usually they kind of think of a different Gaston. And so again, kind of by learning this 1D structure though, we can learn these quote unquote topographic maps of a tissue slice, where you have these colors that tell you this isodepth coordinate, and these arrows give you these directions of maximum increase. And by having this isodepth coordinate, you can find how your tissue is organized by these different contours. And by looking at your measurements along this 1D axis, you can see these gene expression patterns.
Isn't the assumption of the 1D nature constrain the amount of heterogeneity in the sample of cell type?
Yeah, it definitely constrains like exactly what tissues you can look at with this 1D coordinate. So I will discuss in the end some of our approaches for relaxing this assumption. But it is true that if you have a tissue that's like very heterogeneous, in some ways you're assuming that there's a smooth function. You're somehow assuming there's a smooth function f of xy that can describe your tissue. And if that's not the case, if it's not very smooth in space, this approach, these assumptions would.
Is it mostly designed for neural tissue or brain tissue? Where that assumption is?
We've tested it mainly. Well, I'll show one on a cancer tissue. We looked at a few tumors. Here, you do have to assume though that there is like most genes follow like a big gradient direction. We have actually, I'll show some pictures. We also looked at the liver with it. There are a lot of tissues in practice that kind of are organized spatially. But I could imagine, for example, for like a very heterogeneous tumor where it's like very mixed together, this kind of approach would help.
While we're talking about, could you comment on how you would train this?
Yeah, so we trained this, I guess, on given like a data set, like a single tissue slice, we'll train this model like individually per tissue slice. And the idea, I guess, is that each cell that you measure is kind of a training point.
Yeah.
Like the XY is the input, and then the gene expression is the output. And we train it kind of using standard like back propagation. These are just parameterized by MLPs. It's like the simplest architecture we use.
But every sample gets retrained from scratch.
Yeah. So we are working on approaches now to kind of look at multiple samples at once. But you do have to assume that in these multiple samples, there is some like shared structure. I still sort of think of it like an autoencoder in the sense that like if you're doing like PCA on your data, for example, you have to recompute the PCs for like each data set that you look at. Yeah.
So I'm wondering if in some of these cases where you can't like restrict them down to 1D, maybe it's because of the source of the information, because it looks like a lot of this is sections, right? So if you're looking at sections, it's one orientation in one slice, where if you, for example, did I forget the word, but it's like this, that slice, it would look entirely different. And then maybe in that direction, you could find a one-dimensional, or maybe in a complex, in a more complex tissue, if you looked in 3 dimensions, you could maybe find a radial conformation that you could describe in 1D using just radius from a center point. Is there any thought to look at maybe, you know, in any given lobe in a complex tissue, if you look in 3D, with the center being the lobe, and the one dimension you're looking at being the radius from that center, could you then compress that down to 1D and maybe try to get more of these complex tissues?
Yeah, that's a good question. It definitely, yeah. So I guess your question is like how, some ways, how does the slicing of these two, like, because it is a 3D organ. So we're slicing it in 2D. And so maybe there is like this function, it's radial when you look in 3D. But if you slice it at a weird angle or like something, you might not see that 1D structure. That's definitely a real issue. It would be cool to have such an approach work in 3D. I think the collection of like 3D spatial transcriptomics is like very hard. I think there's a few technologies that do it, but mainly what you do is you'll cut like serial sections and try to glue them together. And even that's like a non-trivial problem. So I guess I agree that that's an important problem. I just don't put like the state to think about the 3D model. Yeah.
Well, sorry, but following up on that similar thing, does the assumption of unidirectional, or in this case bidirectional, gradient holds true in greater dimensions? Like, can they all be in the same direction at that point?
That's also a good question. I don't have enough experience with 3D to know, but I think it would have to depend a lot on which tissue you're looking at. Certain regions of the brain. I think it's very tissue specific.
I guess like I was gonna ask, at the beginning you have to assign special coordinates in some way. Is your model like invariant under certain kinds of transformations in these coordinates? So like I like swap X and Y or apply some like linear transform to them. Will your model be like invariant to these kinds of transformations?
That's a good question. Probably not. Because you train this, I guess, on like every single point of the tissue. So the assumption, I guess, is like different points in space, like X, Y versus like X prime Y. You probably will have a different gene expression. So I think if you took the whole tissue and I guess shifted every spot linearly,
Yeah, that's
then
fine.
yeah, shouldn't be. But then I think it depends on like how well you actually finding a minimum. Maybe it could be the case if you like swapped coordinates or did a linear shift, the neural like you don't convert, you converge to a different local minimum. I haven't seen that in practice, but I could imagine if you did a complicated transform.
So the output of this neural network is gene expression, right? And then gene expressions are like sparse, and it's like you have really high dimensionality. So I'm wondering like what kind of did you do any special techniques to address this issue? And like depending on the loss function you use, like the difference between the output might not be that.
Yeah, that is a very good question. Also something I kind of hit in here, but in practice actually, so you could use all genes with like a Poisson or negative 5 neural loss since it counts. If we do that, it tends to be kind of slow to converge. And in practice, what we do first, we use something called like GLM PCA to get these like PCs, and then maybe with 10 or 15 PCs, then you can actually use like a mean squared error loss and like it's a neural network. There won't be as many features, so it can converge. But those are all like very important points. And if you try other feature reduction ideas, so you have varying results. I think we found that we really, at least for these very sparse data sets, you need like a count-based approach for doing dimensionality reduction. But yeah, for example, if you use like the PCs you get from like Seurat or ScanFi or something, it tends to, even if you just visualize them like spatially, they're not coherent.
So when you approximated those functions using neural networks, did you like try to go back and see if D is actually unidirectional as you assumed before when you were trying to find the function?
So in practice, the data is very sparse. It's hard to eyeball. I'll show some examples, but in general, it would be nice to also have an approach to like verify that there is a 1D coordinate or like a hypothesis test to test for whether these assumptions hold. That's something we haven't done, but it's also like a good idea. I think in the interest of time, I'm going to move on because I want to also present a whole different project. So sorry, I appreciate all the great questions. Okay, so this is this cerebellum tissue that I've shown from before. We're using this kind of global model. You can learn this 1D structure that like this 1D coordinate that gives you the kind of layer depth. It's like 0 on the outside and very large on the inside. And these gradients tell you like the directions that expression is increased, just kind of from like the inside to the outside. And by using this topographic map, you can find these different clusters, these different layers. And the kind of cool thing also is that for most genes, they tend to be very sparse. So for example, here's like a gene, which is supposed to be like a node marker gene. And it's like really hard to see, so maybe it looks especially sparse. But like, if you try to look at it, there's a few like dots of red, but it's like mostly zeros. But by aggregating your gene expression along these isodepth contours, you can see this 1D coordinate. You can, or this like expression has this gradient in this layer It kind of jumps, it increases, it decreases. And what's cool also is that by using these patterns of like gradients and these big jumps, you can identify these spatially variable genes, these like important biological genes more accurately. Here, just to show a few more pretty pictures, here's another region of the brain called the olfactory bulb. It does have this concentric kind of layout. And we learned this 1D coordinate that again smoothly varies. And what's cool is that gradients tell you actually trajectories that neurons take. They move from the inside to the outside. I guess these are the negative gradients. But these gradients can't be found by many of these local methods, which don't take into account this global 1D structure. And then here's one more example. This is a tumor. Here, it kind of, this neural network learns with 1D coordinate that again varies from the outside to the inside. And using this coordinate, you can look at patterns across different genes that tell you about things like metastasis or metabolism. And so along the lines of several of these questions, I've been thinking more lately about approaches for extending this model by relaxing these assumptions. And we worked on some ways to relax this assumption by allowing for multiple gradient fields. So subsets of genes share different gradient directions, and also for allowing this isotype function to be kind of discontinuous across these different regions of the tissue. And this is in collaboration with some chemists and neuroscientists at Princeton. And then more recently, we've been thinking about how to use this model to identify cellular interactions. And so we've developed approaches based on Granger causality for finding interactions between ligands and receptors along this isodepth coordinate that can tell you which cells are interacting. And then separately, we've also developed approaches for identifying local interactions between nearby cells using kind of count-based correlation measures. So to summarize, and so to summarize this line of work, we developed a global 1D model of gene expression that you can fit either through a supervised approach using tools from complex analysis or through this unsupervised deep learning approach. And through this, we actively worked on different applications and extensions of this approach, and very excited about this. It's something I plan to continue working on in the future. And so now shifting gears a bit, sorry, it's a bit rushed, but I want to discuss another line of work, kind of shifting gears almost entirely. Instead of thinking about spatial data, let's think about networks. This is work that was joined with, again, many wonderful co-authors, including co-first authors Matt and Tyler, and my PhD advisor, Beth. And this work is on anomaly detection network. And so, okay, I've been talking about spatial data for a while. And in spatial data, one problem that you might be interested in solving, in addition to finding these gradients and interactions, you might be interested in finding anomalies. So for example, if I have data from a tumor, these anomalies, these regions of space with accurate measurements might be a cancer, might be like the tumor itself in this tissue. People also study anomaly detection in space and many other problems. So for example, in epidemiology, you might be interested in these spatial anomalies, which tell you disease outbreak regions. More generally, you can think of these spatial anomaly detection problems as a special case of a network anomaly detection, where you have a network, this graph, that's like nodes or points in space and edges connect nearby points. And you have these scores that tell you how aberrant each node is. And you want to find these kind of subnetworks that are anomalous. And so this is an interesting question in the context of spatial data. But when I first started thinking about this problem, kind of at the beginning of my PhD, we were interested in this network anomaly detection problem in a different setting. Here, we were quite interested in protein and protein interaction networks. So this is a type of graph where you have nodes that are proteins and your edges tell you which proteins interact with them. These large graphs with like tens of thousands of. And so this problem of anomaly detection in protein interaction networks is something called the altered subnetwork problem. It goes by other names like network modules or disease modules. But the general problem formulation is that you have a graph, this protein interaction network, you have vertex scores on each node. And these vertex scores tell you, in the protein case, how much each protein has been altered by a disease. So for example, you can get these measurements from differential expression or GWAS or many other types of data. But the goal is that you want to identify these high scoring subnetworks of your network, these so-called altered subnetworks. So this problem of finding altered subnetworks has been very well studied over the past like 20 to 25 years. These altered subnetworks tell you, for example, groups of interacting genes in cancer. They tell you groups of He's interacting proteins also in other complex traits like diabetes or height. And over the past 20 years, there's been tons of methods for solving this. And so it might seem like this is a very well-studied problem. There's not much to do here. But it turns out actually that many algorithms have this one fatal flaw. They output really large subnetworks. And this is because many algorithms are based on this very seminal approach by Heidegger et al. Called J-acted modules. And they use this scoring function that consistently results in subnetworks that are large and difficult to interpret biologically. And so I read this, I guess, in 2018, around the beginning of my PhD. And I was really curious when I saw this, what this method J-acted modules was doing. Why is it finding these really large subnets? Maybe it's like some really complicated method. It turns out actually though, if you dig into this method, it's doing something that seems kind of simple. It's finding all connected subgraphs S, that have the maximum vertex score divided by the square root of the size. It's a very simple function. And so maybe the sets were really bad. And so I tried to investigate this, but it turns out actually that even in simple simulations, this approach from J-acted module actually has many issues, has a big issue. So here I took if you take a network with 10,000 vertices, and I just implanted a single subnetwork in here. It has 500 vertices, so 5% of the graph. And I give the scores of this implanted network scores that are 2 standard deviations larger than the average score in the network. This seems like a pretty simple simulation. But if you run this approach, J-acted modules, it actually finds a subnetwork that has 2,500 vertices. That's a quarter of your entire graph, and a 5 times increase. And so in practice, actually, what many methods will do is they'll find this connected subgraph and have a bunch of heuristics post hoc to reduce the size of this network. But the effectiveness of these heuristics can be unclear. Yeah.
Sorry, in the example, did you only perturb the scores of the vertices in A, or did you also perturb their neighbors?
I picked a subset A that was like a connected subgraph.
And everything else was like the same score.
And I'll describe the generative model in a few slides, and I'll hopefully give it. Yeah, so in this line of work, our contributions were we defined this generative model for altered subnetworks. And we show that this issue of identifying large subnetworks is actually due to a fundamental statistical bias in these methods. And we developed some approaches which address this bias and are actually asymptotically unbiased. And we later developed approaches that generalize this to other anomaly detection problems. And so very briefly, I want to describe this generative model. So this is something we call the altered subnetwork distribution. And here you have a graph, and you have a single subnetwork A, just for simplicity, that we call the altered subnet. And of course, you can generalize this to multiple subnetworks. But we model vertex scores as coming from 1 of 2 different Gaussians, either one centered at 0 or one centered at some higher mean mu, depending on whether the vertex is in or is not in the altered subnetwork. And so with this generative model where you have these 2 Gaussians, you can pose the altered subnetwork problem as the problem of given a graph and these vertex scores, you would like to find this altered subnet, the collection of nodes that have a much higher vertex score. Just to give a bit of intuition, the difficulty of this problem depends a lot on this parameter mu, how separated these 2 distributions are. If mu is very small, these 2 distributions overlap a lot, and it's really hard to solve this problem. And if mu is very large, then these 2 distributions are really far apart. And in this case, you don't even need the graph. I just do a cutoff right here. And so the interesting cases are really in the middle, where mu is not too large or too small, and you need this graph and the vertex scores to find these subnetworks. And so when you have an estimation problem, here we're trying to estimate the parameter, this altered subnetwork of a distribution, one of the most natural things is to compute a maximum likelihood estimate. So the first thing that we're able to show, which is kind of cool, is that this maximum likelihood estimator actually has the exact same form as this J-acted modules method. And so this is kind of nice because this paper by Heidegger et al., nor any of the subsequent follow-ups don't describe this generative model or this connection to maximum likelihood estimation. So kind of under the hood, they're doing something statistically reasonable, even if they don't know it. But the major issue here, though, is that even though they're computing an MLE, this MLE turns out to be a biased estimator, in the sense that the size of the MLE is on average larger than the size of this true altered subnet. And we actually found this both empirically, so this is kind of the simulation I'd shown before, but also we were able to prove that asymptotically, the limit as your number of observations goes to infinity, this MLE will always be biased. And this is true no matter what this mean mu is. And so this is kind of striking because it gives this theoretical explanation for why these methods for finding these very large subnetworks. And so a natural question then is, how can you reduce the bias of this maximum likelihood estimate? And here the key idea is that you can first model the vertex score distributions before using the network. And using this vertex score model, you can estimate the size of this altered subnetwork, which is given, in this case, we use a Gaussian mixture model because we have these two different Gaussians, and this mixture parameter gives you the size of these altered subnetworks. And so the punchline is that if you use this GMM approach, you actually get a much less biased estimator of the size of the altered subnetwork than using the MLE. And we show this empirically, and we also prove this in a follow-up paper. And here, this is actually kind of interesting though, because it's a case where model misspecification, you have data from one distribution, and if you fit it to a separate distribution that's GMM, you actually get a better estimate of your parameter compared to using the true distribution. So this is a bit striking of the result.
Is that bias a percentage of the size of the network?
Yeah, but it's like an additive term. So like here means, I guess, the network has size like kx, then it's like the MLE is size k plus 0.2. Yeah.
Does the GMM converge to the same, converge the same way as the MLE? If your number of samples is large enough, I guess.
So even as the number of samples goes to infinity, this MLE will still be larger than this true parameter. Whereas the GMM, as n goes to infinity, to show that this alpha parameter converges to the true size. So like asymptotically, there's still a difference. And then we put these insights in an algorithm that we call netmix for finding these altered subnetworks, where the idea is that you first fit your scores to this Gaussian mixture model to find these two components and the size of the altered subnetworks. And you also get something called a responsibility, which is kind of roughly the probability of a vertex belonging to this altered subnetwork. And then you can find the connected subnetwork using an integer linear program that has the maximum total responsibility, while its size is also what you estimate from this mixture model fit. And so, like in most bioinformatics papers, we show that on simulations and real data, our approach does better than existing methods. Here in simulations, we can show that this blue line, which is us, we're much closer to the true size of this subnetwork compared to this G-active modules approach, which is much larger, and other approaches. And that by correcting this bias, we can more accurately measure this subnetwork. And in real cancer data, we could show that by correcting this bias, we more accurately estimate, infer known cancer genes than existing methods. And then kind of in follow-up work, we generalize this approach to something called netmix2, where we relax the assumption that you have two Gaussians. Instead, you have this kind of non-parametric mixture model through something called local FDR. And we also allow for more subnetwork topologies beyond connected subgraphs. So for example, subgraphs that are edge dense. And we also define a family of subgraphs based on random walks, which is kind of what people use in practice to find these altered subgraphs. And we show with this more flexible approach, we can improve identification of disease genes, both in cancer and other complex traits. I also briefly want to mention that in addition to finding anomalies in these graphs, we can also infer these graphs. I've developed approaches for learning gene and protein interaction networks from mutation data, and also approaches for random walks. So these are kind of like aside problems we've worked on graphs. And then finally, just to come back full circle, we also extended these anomaly detection approaches to other structured anomaly detection settings, where you might not just have graph structure data, but you can have matrix structure data, or time series structure data. And the MLE is often used in some of these approaches, and we show that this MLE is biased depending on the size of the number of possible anomalies. And so this is a kind of technical result, but one thing that's kind of interesting is that for spatial anomalies, this MLE turns out to be biased. And so the spatial scan statistics, what's often used to find these anomalies in space. And if you use the network version of this, you'll find these like disease outbreak regions that are much larger than you would expect. It's still not really clear why you would find a region that looks like this inside Manhattan, but it's still much smaller than what like the scan statistic approach would tell you. And so finally, to summarize, in this line of work, we developed this generative model for altered subnetworks, and we showed that the MLE for finding these altered subnetworks is asymptotically biased for connected subgraphs and for other structured anomalies. And we developed some asymptotically unbiased algorithms for finding these subnetworks. And more recently, I've also developed approaches for inferring these networks from data. And so finally, I just want to conclude by describing some of the work I'm interested in the future. So in my research, very broadly, I developed, as I've shown a few examples of, new computational machine learning methods for biological problems. This includes problems in network biology that I previously discussed, like anomaly detection or inferring networks, and also problems in spatial biology using tools like complex analysis or deep learning. Going forward, I'm really excited about these new spatial biology problems and the new technologies that we have to look at biology in space. And so today I discussed some methods for a type of technology called spatial transcriptomics, where you measure these different genes at different spots inside the tissue. And this kind of became popularized at high throughput maybe 5 to 10 years ago. But now there's many new spatial technologies that are starting to come out. For example, you can now do multimodal measurements, where you measure not just genes, but you can measure genomics or proteins or even metabolites at different locations in a tissue. You can even do this kind of within a cell also. There's also now perturbation data, where you can use something called CRISPR to induce these like knockouts inside individual genes. And you can try to infer these like spatial gene interactions. At the same time, though, despite this kind of new, all these new spatial technologies, many of the classical spatial methods tend to be designed for small or low dimensional data. For example, you have point processes or scan statistics, which assume you kind of have one-dimensional data. And so for this new next generation of these spatial technologies, we really need new computational and machine learning methods, which is what I'm interested in developing in the future. And so to give some very concrete directions, one set of problems I'm quite interested in is understanding these interactions across space and using different modalities. For example, can you infer these multimodal networks both inside individual cells and across entire tissue? And using these new multimodal and different technologies, can you also identify these subnetworks, they're so-called niches, that are regions of space that occur throughout a tissue and are associated with poor health outcomes like cancer onset. Another important problem is with perturbation data. An open problem is being able to robustly estimate the effects of a perturbation. So understanding how these gene perturbations affect not just an individual cell, but also how they propagate out spatially. And for example, this could involve developing new approaches in things like dynamical systems or causal inference. And I want to conclude that by noting that in my future research, I really want to develop the next generation of spatial machine learning for biological applications. And this will involve developing both new spatial and network machine learning methods, for example, using tools from, say, PDEs or physics-inspired neural networks, and also using these methods to really understand complex diseases like brain disorders or cancer. And so with that, I'd like to end. I'd like to acknowledge my many co-authors who've made this work possible, including my PhD advisor, Ben, and his group, and also various funding sources. And with that happens.
Great talk. I had a question. You have two methods that seem at first glance potentially disconnected, but have you considered maybe varying them in some way? Like, from my perspective, it feels like there's probably multiple vectors that are probably superimposed. You could group genes as forming patterns in different directions, but if you only look at those genes, it might behave as one direction. Do you create a graph of genes that are in terms of like majority contributing to one type of direction versus the other, and then use your other subgroup analysis to get a better estimate of maybe we should look at these genes and then apply Gaston to these different subsets and then iterative.
Yeah, those are really good ideas. We actually just recently submitted a paper on spatial anomaly detection, spatial transcriptomics.
Exactly.
To combine some of these ideas. It's not quite as sophisticated as what you're describing yet. Pretty sure I have an email talking whether the paper got in or not that came like an algorithm. But no, those are like something I'm also very actively interested in the future, being able to kind of detect these niches, for example, but consider them in space. There should be ways that use this like spatial transcriptome, this high dimensional data to find these different like subnetworks. Yeah, there's good suggestions. Yeah.
So when you're breaking down the cell, looking at the tissues into individual layers, a lot of what you're doing is like you're predicting based on active coordinates, but you're assuming, from my understanding, the homogeneity in cell types. How's that dealing with like intra-tissue layer differential cell types?
Yeah, that's a good question. So one of the ways you can get these gradients is actually through gradients in the proportion of cell types. See if I have it. It shows it. So for example, this is the cerebellum where you can actually look at like different cell types, how they vary in space, and you see like for some genes, there's actually kind of like a gradient of the cell type proportion. And so this gradient we're capturing for many genes is likely just due to the gradient in the cell type proportion. In fact, you can try to distinguish different genes based on whether the gradients due to gradient proportion or like a gradient that seems to be dependent on proportion. But in general, yeah, it would be nice to have ways to kind of account for these different cell types, find cell type specific gradients. If you have accurate measurements of cell types, you could imagine running this approach individually on each cell type. But if you don't know what your cell types are, then I think that's also still active. Yeah. Thanks. Yeah.
How do you reliably call the cell type when they're spots, right? So they're not actually individual cells. And so you're assuming that the whole spot is one cell type, correct?
Yeah, so I think in slides, each spot has only one or two cells. So
Okay.
we actually just used the calling that they did in the group that published this HM's group. They like called each spot based on like which cell type was in that spot. I think if you have spot-based data like Visium, it'd be a bit harder to kind of find these like cell type gradients because you also have to infer for each spot what proportion of cell types are inside it. So I think this approach, it really only works on like near single cell resolution. Here's like another example, just to show another pretty picture. This was like a Visium data. A Visium is one where each of these spots is like a known cell type. So you can look at these different patterns of cell type proportion as it varies along this one equal. But yeah, I guess to answer your question, it's still, depending on the technology, it can be easy or hard to call cell types. I think that's still like a hard problem.
As you said in your talk, biology is driven by cell-cell interactions, but in Gaston, you entirely operate on the XY coordinates of the cells rather than any sort of direct neighbor interactions. And I'm wondering if you sort of considered incorporating graph structure into your model as some other spatial transcriptomics models do, or if not, like sort of the intuition on why the coordinates are sufficient for that sort of prediction.
That's a good question. So that's something we've thought about, and I think it'd still be useful to look at graph structure data. I think with the coordinates, it really helps you find these global patterns, these like global gradients that, like with respect to a single coordinate system that you might not get in graphs. Because with graphs, I guess you know where each cell is with respect to its neighbors, but you don't know its location inside the global tissue. But I could imagine there being like a future approach where you also want to find these global gradients and these local interactions, or maybe. And for that, I would imagine anytime you want to find local interactions, I think a graph's a great way to represent the data. So I think that's, again, another like interesting future direction about how to find local interactions and global structure.
Thank you.
All right, any other questions? Right, the second. Yeah. Are you also? I should, actually, I totally advise them to add closer.
I think if
Yeah, it was before, I said, hey, you learned that the folks in Marble did, and yeah.
that's your paper.
Oh, thank you.
No.
Thank you.
Yeah, awesome.
Are you using the model?
Yeah.
Are you doing so good? a lot of work that you think. Okay, I'll see you tomorrow.
Yeah, been running.
So.
Yeah, of course. Yeah, I appreciate it.
I really, but overall.
No, you're right on that.
I'm really interested in the topic, because I'm a new mathematician, maybe like really. If I know, I will have very enough stories. But I'm very other. Like the slide should be like here. Now like.
Yeah, I want to say my thesis slides are on the site, I'll have to double check, but
Oh,
these are like largely my PhD thesis slides, so.
yeah, I gotta check it out.
Yeah, if you can send me an email, I can send you.
Yeah.
We
Yeah.
did record the conference, we did that in. Okay, so they will at least give you the. You should see,
Where can we find that?
we'll send it out in a couple days.
Oh,
Yeah, yeah.
So
Okay.
they want just one. Yeah, sure. So put it.
Sorry, for which?
The gene expression.
Yeah, so we just use like, actually, it's like.
Oh, just MMP. And also you said.
Yeah, like actually just looks exactly like this structure.
That's amazing that it works.
Yeah, it's honestly just like a throwaway thing, there's like, let's try this and see if it works. Oh, wait, if it worked, we spent a year.
Sparse, but. So you mean the real architecture you're using?
There might be 2 hidden layers, I mean, there's, it depends. You can use 1 or 2. There's like a data picker, but it mostly looks
Okay.
like, it's not a deep, like, I guess it's a deep one, it's like shallow.
Oh, yeah. So there are no any convolution operations like that?
No.
Oh, okay. Also, one quick, and based on this one.
Yeah.
So you mentioned the sparsity for data is a challenge. How do you distinguish? Because the sparse data becomes graded some standards. Already graded. Yeah. Yeah.
Yeah, it's a good question. So there are 2 approaches that. One is that the output of this sparse data we use. We do in practice after that, we first take the gene expression matrix, we use something called GLF-PCA, which like, instead of getting all 20,000 genes, gives you a certain type of dimension. So in that case, you will not just. It's not just normal. I already in fact, it's like a sparse count.
Oh, so you mean?
I don't really care about this particular person. I just.
Yeah.
So, in 12th grade, teaching students have to do a senior project. And one of those options you can do is to have a student.
Well, very somewhere in like this, something smart papers to show like the PCs.
That's what I was basically. Yeah,
But there's only like.
every Monday, Wednesday, Friday. So Wednesdays and Fridays, I was fully off. I didn't go.
It's something nice also that.
I just went to.
You can actually, you can prove actually that if you have like.
Monday,
P
Wednesdays,
different
Fridays, and then
layers,
Monday half.
then if you
So
use
those days
2
I'm
times
just
P
picking up
number of
Because
resources on
you
these
just
genes
hang
goes
out.
2P. 2 times
So
P
Wednesdays and Fridays, you
PCs will
just
also
drop
be
off
piecewise linear,
Or I
like
take the
approximately Gaussian.
or VRE, it's like the mark.
So like theoretically, there's like a nice justification.
Junior Mail Express.
GLF.
else.
Yeah, thanks.
Thank you so much.
Yeah.
That will be first.
Awesome job. Make sure to say hi to everybody for me.
Yeah.
And, we're gonna we'll have feedback.
Those VAR. Is that what you're talking about?
That was like a 2-minute walk.
Like in the box.
Yeah, thank you.
Yeah,
And it was- Yeah, thank you so much for.
Yeah.
Appreciate it.
Are you guys awesome?
Yeah.
All right, enjoy lunch.
Yeah, thanks.
Walk over whatever, it's really close, so there's no rush.
Cool. It's like the art museum.
Yeah, it's like, I don't know if you saw it. It's literally that building over there.
Oh, yeah, that's really close.
I think that's actually, yeah, it's the building over there.
Sorry, give me like.
No,
take your time.
just. People walk by that.